In this recording, we're going to go over the basics of bioinformatics, which is based on sequences. So if we go to the biology open educational resource and hit the bioinformatics tab, we get to this page here. And the first thing that we speak about is the file format that we utilize to store information as sequences. We remember that DNA sequences are A's, T's, C's, and G's, and RNA would be A, U, C, and G. But at any rate, we know that if you have a DNA, we know what the RNA is. And for the ease of transferring data, everything is done as a plain text file, right? It makes it very small. And additionally, if we have a double-stranded DNA, all we need to know is one strand because we understand the rules of base complementarity. And so we don't have to have twice as much information if we have one FASTA file. So FASTA, or sometimes people call it Fasta, the FASTA format is, as we said, a simple standardized way to transfer text from one person to another or machine from one place for another. And basically, plain text is going to be using something called a text editor. So if you're in Windows, you might be familiar with Notepad. I don't suggest using Notepad simply because Notepad doesn't do a very good job of this. You'll probably want to get something like Notepad++, which is free. In a Unix-like system, you might use Vim or VI because those are simple text editors from the command line. Also built in are usually something like Nano or Pico, which are usually pre-installed, right, on your system, if you have a Unix-like system, such as Linux or the Mac OS. We also have on the Mac a proprietary software called Text Wrangler, which is now called BB Edit, right? So it's a little bit outdated, but nonetheless, it's free, as you can get it for no cost. There are certain packages that require some money. And if you're on a Mac, then you have built-in Text Edit, which is like Notepad, but slightly better. But still, these things are not the most appropriate tools for using when you're talking about bioinformatics. You should get something that's a little bit more beefier, such as, like I said, Vim, BB Edit, Notepad++, or whatever generic built-in text editor, graphical text editor, I should say, that you have on a Unix-like system, right? Those are usually better because they have some built-in functions that make them better for, say, programming, lightweight programming, and that'll help you out as well. One thing that we have to understand is that the nature of the FASTA format. The FASTA format has a header line or description line, right? And that means that this is not the information that you're interested in, not the sequence itself, but it's the name of the sequence or the description of the sequence. It begins with a greater than sign, right? So here we have an example of a FASTA file, and so we see here greater than made up nucleic acid sequence, which is the descriptor of it. And then on the subsequent line, we have something that looks like a DNA sequence. And on the subsequent line, once again, we have another greater than sign, which is telling us that this file actually has multiple FASTA sequences within them, right? So that is totally recognized as something that you can do. And these days, when we talk about high throughput sequencing and next generation sequencing standards, we have an additional type of file called a FASTQ file, which we can talk about later, but it's made up of, instead of 2 lines, as you can see here, per sequence, it's made up of 4 lines per sequence. And that is due to the types of information that are carried out in it, right? So we have FASTA sequences. And what we can do is understand that since it's a text file, you might have a text sort of ending to the file, right? Or sometimes you have .fasta, .fa, .fna, right? It doesn't matter what the extension is because at the end of the day, it's simply text, right? Text can have any sort of format. And unfortunately, many of you guys are used to using GUI-based operating systems that default to certain programs based on their extensions. So we just have to understand that an extension is simply an ending. It doesn't matter what type of file it is, but sometimes it might inform your operating system of which program to use, and you can always change those as well, right? We also understand that the DNA letters are A's, T's, C's, and G's. And so you can see those are the codes and what they correspond to. And here we have R for purine, Y for pyrimidine, right? So that means for any one of these two or any one of these three in the sequence. K for G, T, or U, M for A or C. These are a little bit more less used, right? But they do exist for certain reasons. And so forth. Another one that you will commonly come across is one that says just N. And N just stands for anything. It means that it may be undetermined, and we don't know what it is, but we know that there is a nucleotide at that location. There are reasons why you would have an N, mostly as a place holder, especially when you can't determine what something is. It still has value to it. And X means that we have purposely masked that space. That means typically when you're analyzing DNA sequences, sometimes when you're looking at a genome, you have all of these repeated elements that are really not useful information for us. So we purposely mask those regions so that we ignore that when we do our type of analyses in bioinformatics, right? Because if we waste time doing analysis on a region where we're not going to get useful information out of, then it's a waste of our computational resources and time. And sometimes you'll have these gaps, just a space, right? That's when you don't have an N, right? Which means that we don't know if there's nucleotide there or there is. However, a gap is just basically informing us that there's a lack of information. And sometimes those gaps are there when we're comparing sequences next to each other. And that's when it has a real important usage, right? So we're going to explore some FASTA files and how you find them or where you get them from. So let's just go to a repository of some sort. So now I am opening up a new tab here, and for some reason I don't seem to have that. Okay, yes, the new tab is open, and in this new tab, what we're going to see is we're going to look for NCBI, the National Center for Biotechnology and Informatics, and we're going to go to nucleotide, right? So here we are, NCBI nucleotide. And so now this is where we have our repository of information. And so NCBI is this great gateway into information that we are interested in. There's a repository called Genbank that houses sequences. I just chose nucleotide as one of the ones. You can see there are many databases here. I'm just going to choose nucleotide. And for some reason, I'm interested in, let's say I'm interested in a sequence of something. So let's say PUC 18. Right? So let's see. I'm going to do search. PUC 18 is a plasmid, right? And so you can see there are many, many, many hits for this, right? So you can see that many, many hits, this many, of course. So it's a little bit extreme, right? This is some sort of genomic clone, and it's 243. It tells you about the lengths of these things. I'm actually going to be do a refinement of this, and let's just try to say plasmid. And if we can't find it this way, we'll find a place where it exists, usually through a company website, right? So we are looking at this. So here is synthetic construct DNA plasmid vector PUC 18, including artificial sequence. That's not exactly what I want, but it's close enough. One thing that I do recognize here is it says that it is 2,808 base pairs, right? That's why I'm suspicious that it's not exactly the one I want, because PUC 18 should be about 2.3. But anyway, we're going to go click on this. And now in Genbank, right? Notice that it's nucleotide, but we're getting a Genbank file, right? And we'll talk about that as well. So here is in Genbank some information, and our Genbank file is telling us a locus name, a length. It's saying it's linear. It's actually not, so we can actually change that later because this is actually circular plasmid. You can see that some of the features in it, it's a synthetic construct. and that's useful. It has some miscellaneous features noted here, such as from this 439 position to 560. It's an internal standard DNA for quantification of microbial DNA, right? So I don't know what this is. That's probably something that is added that is making it a little bit longer than I'm expecting, unless I'm just recalling the size incorrectly, which is fine. So this one is not exactly the best one that I want to look at, but we'll take it anyway. So what we're going to do is we're going to click on this where it says send to, and we're going to send it to a file. And I'm going to toggle this. I'm going to toggle this to a FASTA, and I'm going to create this file. Right? So now I have to tell it where I want it to be, and I have a home directory somewhere. Don't I have sequences somewhere? Yes, I do have a sequence directory. And it's always useful to include a descriptive name, right? So I'm just going to call this puck 18, right? Fast.fasta. And so it is there. Now, what I'm going to do is, I'm going to try another one. Let's try puck 19. It's a similar one, so that didn't like that. So let's get one day, and here we are. Yeah, it did not like that. Okay, so puck 19 plasmid. Yeah, something's really not happy here. So yeah, these are all not what I'm looking for. So, oh, here it is, finally. Let's see, right there. Yeah, 2.6 is closer to what I'm looking for. Let's see if this is going to show us the correct thing, as I'm expecting. So now you can see something more useful about it, and I'm gonna choose this one instead. As you can see, it's actually telling us that it's circular, which is more appropriate, and it's 2.7, 2.6, which makes more sense to me, right? It gives you some annotations about where this came from, who submitted it, how it was published, or whatever. And also, there's some other information about it here that might be of interest and how it was utilized as a backbone for other types of plasmids, right? And then we have the synthetic construct, right? This is still not the best one that I want because I usually want more annotations in here to show you something. So what I'm going to do is, once again, I can toggle it here to FASTA, and it'll show us, instead of this information, what a FASTA sequence looks like. And here it is, right? So this is exactly what I said it would look like. This is the first line is the descriptor or header, and the rest of it is the sequence, right? So what we're going to do is I'm going to save this to FASTA. I'm going to actually write over the one I had before because this one is a little bit better. I'll replace that. We can also toggle the graphics view of this. So let's see what that shows us. And yeah, it's not loading because it's taking a long time. Yeah, so here it goes. And so this is the graphical view. It's just basically a positional map of things. And for this one, it's not super important, right? This is basically like a genome browser type view, which we can talk about at a different time. So we're going to go back to the Genbank file, right? Because the Genbank file, as I stated, is informing us of other things. It's another type of file that is from the NCBI and has become somewhat standardized and is also useful for having annotations, right? So here we are. We're going to save it as a Genbank file as well, right? And then I'm going to go to a source where I know I have the version I want. It's waiting to save. Okay, so here it is. And once again, I'm just going to call this puck 18. and it's .gb, which is the extension. It's simply a text file that you see here, right? And actually, I'm going to go back here and find the one that I want, which is under here somewhere, where we probably go through this exercise. And it might not be puck 18, it might actually be puck 19, but still it's useful. So it is loading, and as we go down, we shall be able to find, yeah, the puck 19. So puck 19 is going to be close enough, right? So I'm just going to open it in the browser so you can see what it looks like. This is exactly what I want it to look like, and it has a lot more information in it. The difference between puck 18 and puck 19 is they're the same thing, but something is reversed on it. So we don't have to worry about that so much right now. But like I said, we can actually just save this, and I'll just save it over the puck 18, even though I know it's a different thing, right? So those are our sequences that we have. And now we're just going to, once again, explain how these are just simply text files, right? So the way I'm going to do that is I'm going to open up a text my command line. So here's my command line, right? Here we are, and we're going to go to our sequences folder, and you can see those files are there. So I'm going to actually just ask it to spit out this data in front of us, see? And that's the sequence, right? So it's exactly the way we want. Right now it'll scroll off the page, and I can do the same thing with the other one, and you can see there's something different about that sequence, right? One of the things that's different is the way that the Genbank file works is that it has this starter for the sequence, that's its origin. Remember, all the stuff ahead of it up here is just descriptive information. That's not useful in terms of analysis so much, but it is useful for us to recognize certain features of this DNA that other people have analyzed before, or where it came from. And notice that after origin, it starts off in 1, right? So now it has some sequences. And it has these sequences in chunks of 10, right, with a space, and chunks of 10, space, chunk of 10, space, etc., going on until the end of the sequence. And the end of the sequence is these 2 forward slashes. That tells us it's the end of that, right? Now, typically, the way that we look at it is not useful. So when you're using a command line, you have the option, there's 2 commands, one is called less and one is called more. They more or less do the same thing, and I tend to use less. And this way, I can control how fast things are scrolling, right? So here's our puck 19 plasmid, and we can see that basically I can control how much is being presented in front of us, like that, right? So that's useful, right? And I can exit out that. I can also do the same for the FASTA sequence, and there's no reason to use that because you see it fits all in one screen, right? Pretty much. Yeah. So that is the utility of using a text editor. Now, if you look at the size of these files, right, they're not really huge. But as you can imagine, if you have a multi-FASTA file, right, multiple greater than signs plus sequences, then at some point it'll get very, very large in size, in memory consumption. So it's better to use a plain text thing than, say, Microsoft Word, which you're very used to. Because Microsoft Word does formatting things, and that's not what we're interested in. We're just interested in exactly what I'm saying, plain text. So now we're not going to look at it here, right? Now we're going to look at it in a actual text editor, because this is just a view in the command line, right? And as I said, I have something called Vim, so that's the text editor from the command line, and I can look at that FASTA sequence, and lo and behold, it looks exactly the same. Now, this is actually a text editor, because I can actually now go in and change this thing, as opposed to before when I was looking at it at the terminal, it was just spitting out the information for me to view, right? This is editable. I am not going to do that, and I have to exit my text editor like this, right? So now we're going to look at it in a graphical text editor, right? And so I have my graphical text editor open already. That'd be here, right? Now, this is what the text editor looks like. This one is called Zed, XED. This is just on my Linux system, and I have actually just changed the theme because I prefer it to be dark. It's just easier on your eyes. You can choose this one as well. Another thing that I chose to do is I have activated the display of line numbers. You can see there's a 1 there. Maybe you can make out. So I can get rid of that. And it no longer has like this column of numbers that will show me what line number it is. That line number is pretty useful sometimes, so I'm going to add that. And there we are, right? So now I'm going to, in our text editor, basically you can actually do some highlight modes, and highlighting, if you are using some sort of programming language, it might be able to recognize it and highlight that for you, the syntax, which is also very useful. So we're just going to open up our file, which happened to be under sequences, and we're going to look at the this one again, right? So as you can see, we are looking at the oops, I just want to make it bigger. So once again, you have the line numbers on the side, and this seems to be as big as it can get. So so there are line numbers running down the side, and then this is basically something that is graphical in the way that I can change things, right? And what do I mean? It means that I can go like this and start deleting stuff and so forth, right? So this is what I mean by having a text editor. Notice that it's not Word or some word processor. That would be totally unnecessary. This is much lighter weight, and we don't need to preserve anything regarding the formatting of the font and whatnot. You can choose any font that you want to look at it in. Typically, when we're looking at sequences, we want to use what's called a monospace font, right? Monospace font just means that the fonts are all the same, like each character has the same spacing, right? Whereas in other types of fonts, the spacing changes based on which character you have in place, right? So every character is in its own distinct column here, right? That of equal size. So like a capital letter might be larger, might be wider. A W would probably be wider than other fonts, right? Whether it's uppercase or lowercase. You know, things like that, depending on the font also. So a monospace font is very useful as well. So you notice that it's using a monospace. It's defaulting to a 10, right? You can change that as well. So what I'm going to do now is open up, once again, the other file, which is the Fasta. And here it is, right? So once again, it's kind of small. Why is it not working? Control Shift. Oh, there we are. Right? So there, that means I can make this one bigger too, and I didn't even realize it. Oh, helps to press the right button. And so this is useful for us, right? Because now we can see that there are sequences, and if we wanted to change it, right, we can actually get rid of this. Like for some reason I don't want this at the beginning, maybe I'll want it later on. So I don't know. Let's put it like that. It makes no difference. It's just the name, and now I'm gonna save it, and now it has been perpetuated, and that is that, right? This is actually Puck 19 at circular, and that's great. And this was the one I actually want because if you look at who deposited it, right, it is deposited by a company called New England Biolabs, and that's where I'd probably be getting this from anyway, this company. So that's useful. Now, we're going to examine what this looks like in another program, right? And so the other program that we're going to use is one called UGENE. And the reason why I use UGENE, or tell you to use UGENE, right, graphical sequence analysis, and you can actually download it from Unipro UGENE, is because it's just a GUI for all the command line tools that a bioinformatics student would be using from the command line of a Unix system, right? They would be installing those packages individually and running them. UGENE is basically a GUI, which makes it easier for us to work with and see how to deal with these sequences. So I've already installed it, and now I'm going to go to UGENE, right? So here's UGENE, and what I'm going to do is I'm just going to open a file. And the file I'm going to open is in my sequences. And here we're going to look at the Fasta file. Right? And this is our Fasta file, and it has this multi-panel view of this. And as you can see, there's not much going on here. The only useful one is probably this window in the center, right? The window in the center is useful because that's the actual sequence. And you can see there's numbers there, right? And you can see it goes 1 through, and if we scroll down in this middle panel, it scrolls down to 2,686 or something like that, right? But if you look at it more closely, what you will realize is that there are letters above this sort of like sequence line here that tells you the position, and there are sequences below. But if you're very careful, you will see furthermore that it's the top, the letters above the sequence, right? Especially if we go back to our text editor. So our beginning is GCGCCC, and let's go back to UGENE. And above these numbers is GCGCC, right? So we know that the sequence above the sort of position numbering system is the Fasta sequence. So what's the one below it? The one below it is the reverse complement. Notice that in our text file that we just opened, right now, there is no second sequence, because at the end of the day, we automatically know what that is, right? And if you look at it here, we can actually turn that off. See, now it's not showing the complementary strand, right? So this just helps us out a little. We should be able to figure it out. And notice that there is no 5 or 3 prime notation because we by default know that we're starting G is 5 prime, and all the way down here is the 3 prime end of that strand, right? And the other strand at the very bottom here, that T, CT, is the 5 prime end, and the 3 prime end is all the way up here, right? So those are just de facto standards that we have to keep in mind when we're analyzing data, right? So this is useful for a certain reason, but in UGENE we can do other things that will help us identify certain features of this, right? So we don't know this is circular, but it is. And so what we can actually do now is, because we know it's circular, what we can do is actually highlight the sequence here and right click or command click or whatever you want to do, and we can mark this as circular. Okay, so now we've marked it as circular, and you can see it is a circular plasmid as we are expecting, right? This is still not super useful for us, except for the fact that it has a bunch of numbers going around in a circle, right? So one thing that we can do is to start doing some analysis on this. So we can analyze, and we can start to annotate this plasmid, right? One thing we can start to do is, if we click on Annotate Plasmid, it'll basically give you all these options. We're looking for promoters, terminators, genes, regulatory sequences, origins of replication, primer, common primer binding sites, and other features. So let's click on that and see what happens. Hey, nothing happened because we didn't actually have any information anywhere, right? So, oh, there it goes. Sorry, it was thinking. And now you can see this bottom panel has some of the highlights that are located here, right? So that is something that has happened automatically. That's the auto annotate. And now you can see positionally where these things are. Notice that there's something called M13 puck reverse primer, M13 reverse primer, M13 forward primer, M13 puck forward primer, and they're approximately the same area. And those are what we talk about are useful standard annotations, right? of primers. Those are commonly used, and that is specifically used for sequencing purposes, which we will talk about at a much later time, right? We can spin this around. I'm using my scroll wheel to do this because sometimes, there's not enough space to show annotations. However, so far I see a bunch of primer binding sites. I see no genes, right? So what we're going to do now is do right click on it again and analyze. You can also get this up here, but we're just going to do it down here. Analyze, and we can find what are called open reading frames, right? Open reading frames are exactly like they sound. It starts from an ATG until a stop, right? And when we click this, it's going to ask us, what's the minimal length, right, of base pairs? I actually don't like a minimal length of 100, right? Because 99 divided by 3 is 33 amino acids. That's not a real good protein. So I'm going to do a minimal length of more on the order of, I don't know. Let's be a little demonic about it and say 666. And the max result is well, it's not even going to get to this because of everything that's happening there. And then we have the option of checking off the standard genetic code and some alternative genetic codes, right? So the standard genetic code is what we call the universal one, but it's not really universal because of the way evolution has occurred in geologic time on this planet. There are certain organisms and genomes that have went a little bit different, right? So vertebrate mitochondrial gene code is very minorly different. I don't know what it is off the top of my head, but there's also alternative codes and such, right? So start codon of ATG, and in some bacteria, they use an alternative start codon. So we'll just use the standard, right, for now. Notice what I'm doing here. It's asking us about strand, right? So the strand is default click to both strands, right? Remember, there's information going in both directions. Or we can say direct strand, which is the one that's in the FASTA sequence, or we can just say the complement, which is the one that's reverse complement to our FASTA sequence. But it's important for us to mine both. So we're going to put both. It's going to ask us for an output. And right now, it's going to save everything to what's called an auto annotation table, which is what this is, right? Or we can create a new document. We can actually create a Genbank file from that. And that would also be useful, right? So I will actually do that for the hell of it, right? My puck 18, right? Genbank file. So we'll just do that. And so annotation parameters are auto right now, whatever. And so we're just gonna go back here. We're going to let's click on preview and see what happens. So it's telling us some information that it found a gene of this length, 858, on the direct strand, right? So that's fine. So let's see, we can allow overlaps, alternative initiation codons, include stop codon, whatever. So here we go. So let's go press OK. And you can see that there is what we call an open reading frame, right? And that's correct, right? Now, I know that there's another open reading frame here that it's not finding, and it's probably because we were a little bit restrictive in our terms. But one thing that you should notice right away is here's an open reading frame, right? And it is going in this direction as the arrow is pointing at us. And here's another annotation map, right, along this sort of position line. And here's the actual sequences in this panel, right? So you're starting to see why it's useful to have a graphical representation of this as a map. And then the other thing is what we can see here is upstream of this open reading frame is a promoter, right? And so we have this open reading frame here. And one thing that's useful about it is if you click on it, right, you can see it's showing us the size, its position on this map, its highlighted sequence here. So it's actually starting over here, right? And here you can see this is the AMP-R promoter, right? And that makes sense that this promoter is telling RNA polymerase to go in this direction, and this gene happens to be going in this direction, right? And so you can see ATG, right? So that's a start codon. That's also methionine in the universal fashion, right? And one thing that you notice every time I highlight it or mouse over it, it's telling us a sequence, and it's also telling us the translated sequence to some degree, right? So this is another beauty of having this graphical interface because it's doing many things at the same time that we normally would like to do, right? So we found one open reading frame, and you know, we've actually saved it in this Genbank file here, and it's a different Genbank file. It actually doesn't include the sequence. It's just the annotations. So that's a quirk, a funny quirk about UGENE. But there is a way that you can merge the annotation with the sequence file to make a full Genbank file. So just to illustrate what I'm talking about, we're going to go back here to our graphical text editor, and I am going to open up my, oh, it didn't save it yet. That's not useful. So let's see, let's save it. So let's just try export it. Genbank file, sequences, so this is a copy. I don't know why it's not there. Let me see if it actually showed up and I just don't see it for some reason. Sequences. Yeah, it's not there for some reason. Sequences, mypuck 18. So, yeah, let's export that. Yeah, for some reason it hadn't, it's in our project here, but it's not there. So you can see this project is just the copy of this annotation file that we're making, which is separate from the sequence file. So once again, I'm going to open up my text editor and the graphical one, and I'm just going to show you what has happened, right? As I stated before, it's not going to have the sequence embedded in it at the end. So here we are. And look, that's exactly what I said was going to happen, right? So all it says is, hey, it's today's date. There's an annotation, right? And the annotation is some sort of open reading frame, right? It's not really doing the best job. It's calling a miscellaneous feature. Sometimes you'll see the better way to mark it is called CDS for coding sequence. And, you know, it's telling you the length of the DNA and the subsequent length of the protein that's associated. But look, it just goes to the end with these double forward slashes, right? So that's only marginally useful, right? What's really useful is if we can actually save these auto annotations as well, right? So that requires a little bit more work. And so you can see there's some editing annotation stuff. You can expand and split and join annotation parts. We're just going to ignore that for now. And we're just going to continue on to see now the difference between this and what the Genbank file from the company was. And so we're going to open that. And we're going to go to this one, which is actually Puck 19, but close enough. And notice the major difference about this. The major difference is that it has additional information about origins of replication, right? So this is built in. And the other thing is it has another open reading frame here, right? So we're going to click on that and actually see how it is that we missed this. How did we miss this? It has a sequence. Oh, because I was too restrictive. It's a smaller gene, right? And as you see here, there's a open reading frame called BLA, BLA, right? And so let's do a comparison. We go back here. This open reading frame here that we have, right, is called ORF because that's all we found, right? And let's see, switch to editing mode. Right. So if we have here this information, we can see that it goes from 885 to 1742. So we know that's about like 900 ish base pairs, right, for this gene, 900 base pairs. If we go to this one here, we can see that blah, right, this blah 1 goes from 1626 to 2486, and that is roughly about 900, right? So it actually is the same thing as what I'm getting at. If we did additional things like go back here, do analysis, and do this auto annotation, right? Let's just go get rid of some of these because there will be some duplicates in some of the things that we have here. Let's go annotate it. And so it's running a task, as you can see down here with the progress bar. And there it is. And now you see a few additional things. Notice here what has happened. Here we have amp R promoter. It's something that UGENE knows exists, right? So, and we didn't do the open reading frame finder, but look, if this gene is, which is annotated as LAC-Z alpha, is going from 146 to 469, we know that, you know, it's less than 500 long, so we should be a little bit permissive and say, let's just say, let's see, so that, not 500, I should say 300. So let's go back and to our initial Fasta file here, right? And let's do another ORF finding. Find ORF, right? And so let's not be so permissive and let's just say 300, right? Preview, right? And now you see that we have not only the one that we found, but we have two of these short ones that are either going opposite or in the same direction, right? And so we can say okay. And notice that it just added another ORF right here. So let's just get rid of that one, which is remove. Oh, we can't do that here. Okay, in any case, you can see that there is one going in the same direction, right? That there is one going in the same exact direction. And remember what I said before, Puck 18 and Puck 19 have this, there's something reversed about it, and you can see it's doing it like a mirror image type thing. So we know that this one, and if we look here, there's a promoter. So this is the open reading frame that makes sense, because this open reading frame that goes in this direction, there is actually no promoter that's recognized that goes in the same direction. So here is this one. And if we look at it, right, once again, highlighting this one right there, we can just let it mouse over for a little second. And we can see it goes from 216 to 536. 216 to 536 is actually about 300-ish, as we knew before. And let's go back to our this one, right, the Puck 19 one, and we can see that this LAC-Z alpha has this promoter this distance away from it, and if we look at it very carefully, it goes from 146 to 469, which is about 300, a little bit over 300. And it's about in the same position. So you understand what the utility of having this graphical system is to help us analyze from scratch something that we know very little about, right? It hasn't found the origin. We can do some additional analysis about that and choose one of these tools to find such a pattern. And also what we can do is we can actually export these sequence of selected annotation, which I'm looking at here, right? So we're going to do that, and so this is the Puck 18 annotation, so we're going to export that, and it's added over here, right? So that's not super useful right now, but essentially what you can do is analyze this sequence a little bit more, right? So we can actually copy this direct strand, right? That's one option, and that means we can paste it somewhere, right? We can think about some other analyses, and one analysis that we can do is do a local blast. Now, we don't have local blast necessarily installed, so we can query NCBI blast database, BLAST stands for Basic Local Alignment Search Tool, and that will send this information to the server in Maryland, and then it will look amongst that database for similarities to things and yield those back to us. So let's go click on that. We're going to use the one called blast N because it is actually a nucleotide blast, right? And there are different options that we can pick off. So I'm going to click on that. And see, once again, it's asking me which database collection. We can look at NR, which is the nucleotide collection, or we can do what's called some other things like refseq, right? And any other ones. We'll just do this one for now. That's just easier. And it's going to actually look for saved annotation somewhere. So there we go. So let's just go search. And as you can see, the task is at 0, so something's happening. It's a remote task, so we have to wait for whatever happens in on the server, right? But the other thing that you can do is, if you don't have the patience for this, is what you can do is copy the direct strand, as we see here, and then we can actually go back to the browser. Once again, here's NCBI. If we really wanted to, we just go to open another tab, NCBI, and from that we can directly put it into the BLAST database. So we can just look at where's BLAST? It's on here somewhere. Yeah, and when I can't find things directly like that, oh, there it is on the side. Here it is, right? And so this is being done on the side, and we're doing a nucleotide blast, so we're just going to click on that. And what we're going to do is just paste it into this little box that we have here, if it ever finishes loading. So there we are, finish loading. So as you can see, there's some like speed issues with my network right now. So here we go. I'm just going to paste it in there, right? And once again, we can do, it gives us the same options here that we see. So that's fine. And I am going to choose, no, that's fine for now. There are some things that you can change, but we won't because we don't understand how to do that at this moment. And what we're going to do, I usually click off this thing so it'll open up in a new tab. The reason why is sometimes you need to redo it. We're doing a T-blast. Oh, I picked the wrong thing. Here we go. Yeah, somehow that new one got selected. See, and it failed, which is fine. Right? So here we are. And this is where you would see these things. Highly similar, Mega Blast. I did it a different way. I did this one, but let's blast it again. And here we go. Let's see if it finished in UGENE. And it actually did, right? So you can see that it has all of these aligned blast results up there. But I'm just going to go back to show you the what's happening in the browser. Basically, it reloads this page every few seconds because it's waiting to it's going through the database, right? So it's been 9 seconds, and it's going to automatically update in 2 seconds, which it is now. And here's everything that we have, right? So as you can see, here are our sequences. Many of them are in something called cloning vector. And basically, yeah, synthetic vector and all that stuff that we have there. You can go to the graphical summary, right? The graphical summary is in this tab, and that's going to load. At some point it will load. And while we're, oh, there it is. And so this looks kind of similar to what we had seen as our results in UGENE, right? It has just a bunch of bars where it says this is our query, and then these are the results. And once again, you can mouse over them, and they'll say something that we already see. And then the other thing that is useful on the web side is when you choose alignments, because alignments will actually show you what your query is, and what your subject is, and it'll show you the matches and the positioning of the subject that it found, the result. And you can see it's giving me some other information that this is a 100% match, 858 out of 858. And also you can see something here. It says strand plus minus. That means our direct strand is going in the forward direction, plus, and it's matching the reverse strand of this this query here, right? And so if we go down, you can see another one. It's also 100%. There's a secret to this. It's 100% for a very specific reason. It's found in all of these cloning vectors because it is what we call a selection marker, right? So what does that actually mean, right? So you can see all these things, and we can go through all the results, and you can see that it's giving us these information. So let's go to this one. Hey, it happens to be this cloning vector, right? And that's the one that is highlighted right now at the top. And so what is actually happening? Notice that they're all identical in length. So if we go back to our pre-annotated one, the PUC19, which is this one, you can see it says blah, right? And blah is actually just a shorthand for describing what it really is. So let's pull up, resize these panels and show us what this blah is, right? Notice, right, on this version, on this PUC19, everything is on the complementary strand. It's going the opposite direction of the numbers, right? So, and the arrows are going the opposite direction. So that's what it means to be, so that means the information is on not the direct strand that we put in, but the reverse of that. And so if we look at this, it says some information. It's calling the gene blah, but that stands for this product of this protein, and the protein is called beta lactamase. Notice on the very bottom it has the translated strand, right? So these are not A's, T's, and G's. These are the individual alphabetical designations of the 20 amino acids, starting with methionine, right? And as you can see here, what the note says is what the real thing is, right? It is AMP-R. It confers resistance to ampicillin. And that's what we mean by it is a selection marker. This has the sole function of being part of a plasmid vector so that when you transform it into bacteria, you can select for it, right? You know which ones have been transformed, and that's why you find it as a blast target in many, many different vectors and plasmids that were labeled, because they're all engineered and they have the same exact sequence in them for that purpose, right? If we chose to blast this one, you'll also find it in many, many different vectors, right? It is the replication of origin for our plasmid, which is another sign that this is an engineered vector because we need a selection marker, an origin replication, and another thing that we need in an engineered vector is what we have here, and that is labeled here, multiple cloning sites, right? So this, as we can see here, we've actually highlighted over here, and these are a, oops, not there. Multiple cloning site is over here, and the multiple cloning site you can see here, that's here, is actually a cluster of artificially engineered restriction enzyme sites, right? They don't exist there in nature in this gene, right? This gene being called LAC-Z alpha. And if we go to this annotation, LAC-Z alpha will be here, right? And the product is something called beta-galactosidase. And you can see the protein translation sequence there. Now, notice that this multiple cloning site is within this gene product, within this gene, I should say. And so therefore, this engineered site has the propensity to be digested at those spaces, at those places, right? So what does that actually mean? So let's do another analysis. We're going to analyze, and we're going to do find restriction sites. And notice that there are pre-selected ones, because these are fairly common when we're talking about genetic engineering and making recombinant DNA, right? BAM-H1, BAGEL2, CLA1, DRA1. Those two I use less. ECHO-R1, ECHO-R5, HINDI3. Those are fairly common. PST1, SAL1, SMA1, XMA1, right? And so those are, it's no harm to use those by default. We can also find some other ones. These are also what we call 6 cutters, but we can look for an 8 cutter, right? I know an 8 cutter called NOT1. And I don't know if there are any NOT1 sites in this restriction sequence, but you can see here it's GC, GG, CC, GC, right? So that's 8. So I've added it to my list, and so we'll say okay. And now, as you can see, it's found a bunch of these sites. 1 BAM-H1, 3 DRA1 sites, 1 ECHO-R1, 1 HINDI3, 1 PST, 1 SAL1, 1 SMA1, right? And if we look at where these things are, we ignore DRA1. DRA1, it has 3 sites, right? So there's one here, and one here. That is not useful. It's not going to be, because we want unique sites in our multiple cloning site. And so we can see that the cluster here are showing us those unique ones, ECHO-R1, SMA1, SAL1, etc. Right? Once again, we can move this around just to make it a little bit easier. We can zoom in a little as well. And so you can see ECHO-R1, SMA1, XMA1, BAM-H1, SAL1, and HINDI3 are all in this region, right? So that means we can digest this plasmid at that location uniquely. It will, the double-stranded DNA will open up as a linear fragment, and then if we insert a piece of DNA there using similar restriction sites, then they will momentarily come in contact with them, and then you can use a DNA ligase to basically seal it back up, right? So that is something else that we've learned from this cursory study of using UGENE to look at a FASTA sequence and look at the difference between a FASTA sequence and a Genbank file, and also understand the significance of annotation, right? So at the end of the day, FASTA is simply the sequence, a direct sequence that we have in text, and it's very useful for us to utilize that for other purposes later, right? So we're just gonna call an end to this segment where we have used UGENE to look at FASTA sequences, Genbank sequences, understood the differences of text editors versus word processors and analyzing sequences, and additionally understand why we're using various tools to analyze our sequences that we have there.